With the complement system, there are going to be two main pathways that I want you to focus on. The first is going to be the classical pathway. The second one is alternative. There is a third pathway that I'm not going to have you worry too much about for this course. But in terms of the classical pathway, this one is going to rely on an antigen-antibody complex. So this is going to be one example of where the innate immune response is going to need something from the acquired immune response. All right, and what that need is are the antibodies. All right, the alternative pathway is a little bit different. The alternative pathway is going to recognize those markers on the outsides of cells. All right, but I'm going to start with the classical pathway. All right, we'll have, in terms of our classical pathway, you're going to have a pathogen that's going to be circulating. It might be bacteria. Most often it's going to be bacterial in nature. Those bacteria are going to have those PAMPs on the outside that are going to tag it as something that doesn't belong. And what happens, all right, what we've got circulating are antibodies, okay? And those antibodies are going to be proteins that recognize other proteins. And it recognizes those other proteins very specifically, okay? So in terms of that, we get our antibody that's going to recognize the pathogen on the, because of the markers on the outside of the pathogen. Then what's going to happen, those antibodies have a region for complement to bind, specifically C1. All right, so in this case, complement protein 1 binds to our antibody. All right, what that does then is it activates a cascade of events that'll happen. All right, I'm not overly concerned that you know all of the molecules involved in the cascade. I've got some of them up here. All right, knowing C1, I want you to know, because C1 is the one that binds to the antibody, okay? And that's what's going to start this process, all right? That antibody binds to C1. C1 then activates two other proteins associated with the complement cascade, C2 and C4. Those kind of split in half and give us an A and a B. So we've got 2A, 2B, 4A, 4B. 2A and 4B are needed to activate the third complement protein, C3, all right? And this is going to be the one that's going to be important in activating our innate immune response, okay? Because once C3 is activated, then what you've got is you will trigger either the process of opsonization with 3B or cell lysis. 3A can actually be one of the triggers then that starts your inflammatory response as well. So in terms of the complement system, it kind of has a couple different arms that it can extend into the innate immune system to destroy an invading pathogen, all right? So in terms of that, we can trigger opsonization or lysis or inflammation depending on which way we go down, which pathway we go down, all right? And I know I haven't talked about opsonization or lysis yet, and that's what I'm going to do over here, all right? So in terms of opsonization, all right, what we've got essentially, you've got those markers on the outside of the bacteria cell, the microbe, all right? With opsonization, you're essentially talking about antibodies binding all around the outside of our marker, on those markers. Oops, I drew my Y's wrong there, didn't I? We can fix that. All right. So in terms of that process of opsonization, it tags the bacteria and can essentially activate this complement cascade with C1 binding to the antibodies. Or with opsonization, what it can do is it will trigger essentially, draw one off this way, something like our macrophage to go ahead and engulf that particle. Because having those antibodies all the way around the outside of something is a trigger for that phagocytic cell to engulf that particle, digest it, break it down. All right? So in terms of opsonization, we're talking about the antibody coating around the outside of the bacteria. We get things phagocytosed by the macrophage. All right? The other thing that can happen that I have over here is lysis. All right? And in terms of lysis, all right, we're talking about something that's going to be very similar to what happened with the defensins. Remember, with the defensins, we were poking a hole in the cell membrane. The complement proteins, the other complement proteins, and I think it's like complements 5, 6, 7, 9. Don't worry about what order they come into play. I'm not worried that you know that, okay? But the rest of those complement proteins that are circulating, essentially what happens when that C3b is released, that gets those other proteins to come together and they form a pore in that pathogen's cell membrane. So essentially what happens with the activation of complement, it's a little like the defensins in that you're poking holes inside of those microbes. When you poke holes, then the stuff that's inside the cell goes out. When the stuff leaks out of the cell, that's no longer compatible with life, okay? So in terms of that lysis, we're talking about a process very similar to what happened with what I was talking about with defensins, all right? So we've got opsonization, we've got lysis, and in terms of that with our third round here, all right, with that inflammation, all right, essentially what you'll get, you'll get some of those chemokines, some of those signals that are going to trigger the inflammatory response. So you'll get the release of something like, say, a histamine. That histamine then is going to cause the local vasodilation, bring in white blood cells. The white blood cells, like the macrophages and neutrophils, are going to come in, destroy things. And in terms of that, that's another arm associated with complement. All right? So in terms of that, the classical pathway, what's important about that classical pathway, you've got the involvement of antibodies. All right? The alternative pathway is another way that we can target bacteria. But in terms of the alternative pathway, there are some other proteins that bind to those markers on the outside of the pathogen. I'm not concerned that you know the names of them. But what that does then is that's going to recruit specifically complement C3. All right? C3 then is going to, again, it'll split into 3A and 3B. Okay? And in terms of that, the trigger for that is going to be the same as what we had over here with the classical response. Okay? So 3A is still going to trigger inflammation. 3B will trigger our opsonization or our lysis. All right? So in terms of the complement pathway, I don't want you to remember, I have some kind of complex diagrams in the PowerPoint. I have that in there just to kind of give you an overview of what's happening and how big this process is. There are really a lot of proteins involved. I'm not so concerned that you know the function of all the different proteins. The ones I have up here on the board I want you to be kind of familiar with, because that C3 is going to be important in triggering either our inflammatory or the opsonization or lysis. The classical pathway, again, C1 is going to be what specifically recognizes the antibodies. So know those. Know what it means for something to be opsonized, what it means for the lysis to happen. And I think the diagrams I have actually show you how all those proteins come together to form that pore. All right. So in terms of that, we've got the classical, the alternative pathway. There is a third pathway that will activate the complement system. That's by binding a specific protein known as lectin. And in terms of that, when lectin binds, it's going to trigger some of this same cascade. All right? I'm not so concerned you know lectin, but know the alternative and the classical pathways. If you have any questions, please let me know.